which you have read in the earlier semesters. So in the microbiology, there will be the microbes which you have to be studied, right? As the name suggests, microbiology is the study of microbes which are not visible by the naked eyes and only visible by microscope, right? So inside the microbiology, there are multiple types of the microbes such as bacteria, virus, fungus, parasites, algae, right? So the, for example, the tree is microbiology is a tree. Microbiology एक tree की तरह है. The microbiology is a tree and the parasitology, mycology, serology, and immunology. So these are the branches of single subject which is microbiology, right? So inside the parasitology, you have to read about the parasites, right? You have to get the detailed knowledge of parasites, laboratory diagnosis of the parasites, life cycle of the parasites, and symptoms of the parasitic infections, like that. And same as in the virology, you have to get the detailed knowledge of viruses, right? What are the viruses? What are the properties of the viruses? What kind of the disease are caused by the viruses? Just these concepts. And the most important thing, as a technician, you have to know that the lab diagnosis, because all of you have to work inside the laboratory. So that's why laboratory diagnosis is very important for a lab technician, right? So there are different types of the laboratory diagnosis depending upon the type of the microbe, right? For bacteriology, for bacteria, there will be the different lab diagnosis. For parasites, there will be the different, and for viruses also, there is the different laboratory diagnosis. So for each microbe, there will be the different kind of the laboratory diagnosis, because each microbe cause different type of the disease. So depending upon the nature of the disease, depending upon the symptoms, the diagnosis will different for each microbe, right? So just in simple words, parasitology stands for the study of parasites, right? And virology stands for the study of virus. So let's move on the next slide, in which we get the detailed knowledge of parasitology. That, as I already told you, that it is the branch of microbiology. It is not a different or unique subject. It is a branch of only microbiology subject that concerned with the phenomena of dependence of one living organisms on another. Basically, in parasitology, we have to deal with the parasites, right? And what are parasites? Parasites are those organisms that require a host organism for their survival, right? Without a host or without another organism, a parasite is not to survive. So what is parasite? Parasite is a kind of organisms which require another organism for their survival. Is it okay? So it is a kind of the subject in which you have to get the detailed knowledge of those organisms which are dependent on another organisms for their survival, for their for meeting the nutritional requirements for their habitat and more. So parasitology basically deals with those parasites which infect the man, because we are only studying those parasites which causes diseases to us, right? Because we have to find out the solution for that particular disease. That's why we are reading the detail of those parasites. So in medical parasitology, we have to only deal with those parasites which infect man and the diseases they produce and the response generated by him, means which kind of the diseases they produce in our body and what symptoms they can arise in our body, right? And then to find out the various diagnosis methods, how we can diagnose them, either it is a parasitic infection, viral infection or bacterial infection. So it is depending upon the diagnosis which is done in the lab, right? So after diagnosis, our motto is to prevent the disease. So that's why we are reading or we are getting knowledge of every subject, because ultimately we are the lab technicians or we have to find out the diagnosis of the particular disease, right? So medical parasitology is a subject in which you have to deal with the parasites which infects the man's and uses the diseases, and also you have to know the response generated by him, and you also aware about the various methods of their diagnosis and how we can prevent them, right? So this is about the parasitology. The next, what are parasites? So as I already told you that a parasite is a kind of the organism that lives on another body, and that another body is known as the host, right? So there are two terms. One is parasite and other is host. So the parasite is that organisms which is living or which is dependent on another, right? This is the parasite. And the organisms which harbors the parasites, right, and provide nutritional requirement, provide shelter to the parasite, this is known as the host. So the definition is very simple, that a parasite is organisms that require a host to get it foods or at the expenses of the host, right? So there are three main classes of the parasites that can cause diseases in the human. So we have three types of the classification of the parasites which can cause diseases in the human. First one is the protozoa, second helminths, and third is ectoparasites, right? Protozoa, helminths, and the ectoparasites. This is the classification of parasites. So let's get a detailed knowledge of classification. That parasite basically broadly classified into protozoa and helminths, also ecto and endoparasites. So just understand that what is the difference between endoparasite and ectoparasite. So as the name suggests, endo means inside. So those parasites which living inside the host, inside the body of the host, that are the endoparasites, and ectoparasites are those parasites which can living on the surface of the host, right? Which can live on the surface of the host, body surface of the host, that are the ectoparasites, right? For example, lice, right? So broadly, a parasite is divided into two classes, protozoa and the helminths. So what is the difference between protozoa and helminths? Why they have divided into two classes? So the difference is that protozoa is a unicellular organism, right? It is made up of a single cell, while helminths are multicellular. They have the multiple types of the cell, right? And also they belong to kingdom Animalia, while protozoa belong to the protists. Then protozoa are further divided into four types: amoebic, flagellates, sporozoas, and ciliates. So what are the differences between amoeba, flagellates, sporozoa, and ciliates? So the difference between these protozoal classification is that the amoeba is a kind of the organisms which have no shape, right? It is a organism which is made up of a single cell, but there is no definite or fixed shape for amoeba, right? And second class is flagellates. Flagellates are those organisms which have flagella on their surrounding for their movement, right? And sporozoas are those which can form the spores. And ciliates are those one which having the cilia for the movement on the surface of the parasite, on the surface of the protozoa. So example of amoeba is Entamoeba histolytica, Entamoeba nigraria. These are the examples of amoebic protozoas, right? And examples of flagellates is Giardia and Trichomonas. An example of a sporozoa is Plasmodium, Toxoplasma. An example of ciliates is Balantidium, right? So just remember that this is a very important, that if someone asks you that what is the example of flagellates, so you have to answer Giardia species and Trichomonas species, right? So just note down these examples, which protozoa comes under which category, right? Because there are different type of the laboratory diagnosis also on the depending on their categories, on their classes. And next is helminths. As I told you that helminths are multicellular and comes under the kingdom Animalia, right? So helminths are then further divided into three classes: nematodes, cystodes, and the frammatodes. So the example of nematodes is Ascaris species and Ankylostoma. We will get the detailed knowledge in the further slides. So just note down the names, that Ascaris and Ankylostoma is a nematode, and Taenia and Echinococcus is a cystode, and Fascicola and Schistosoma is a framatode. So this is about the classification of parasites. So after classification, just see or just understand the basic laboratory diagnosis of a parasitic infection. As a lab technician, you have to get more knowledge about the laboratory diagnosis, as I already told you, because you have to diagnose the infection causing agent in the sample of the patient. That's why laboratory diagnosis is of great importance. So first of all, for laboratory diagnosis, the first thing is you have the specimen, right? So collection of the specimen is very important, plays a very important role in your testing, because if the collection of the specimen is wrong, if the procedure if the procedure followed for the collection of a specimen is false or is wrong, then ultimately the result will also get wrong, right? And your report will be false positive or false negative. So to run the test or to run the sample smoothly, collection of the specimen is very important, right? So first of all, collect the specimen in a wide mouth container without contamination of urine, water, and any other disinfectant. So students, just write in the chat box that what is the specimen for parasitic infection. I just want to check your knowledge that which kind of the specimen is sent to the laboratory for identification of the parasites in the sample. Just write in the chat box. Stool, majorly stool, because parasites are those, mostly parasitic infections are related to the GIT tract, that is the gastrointestinal tract, right? So that's why stool is the major specimen which you have received inside the parasitology laboratory. So first of all, depending upon the specimen, ask the patient and give them to general instructions which the patient have to follow while collection of the specimen, right? So first is ask the patient to collect the specimen and instruct accordingly. First, if the next is if the specimen is fecal, if the specimen required is a stool sample, so 2 specimen is obtained on successive days during normal bowel movement, right? And third is after magnesium purge, or the liquid specimen of stool should be examined within the data of 30 minutes. And semi-formed stools should be examined within 60 minutes, right? After the collection of the stool specimen. Because some of the parasites are not, if the parasite is in the living form in the sample, and you will observe the specimen after 60 minutes or after 2 hours, so there will be chances of the loss of the parasitic material in the stool, right? That's why the diagnosis will be made accordingly and timely. And blood samples, like you know what is blood sample. So blood samples are also received inside the parasitology laboratory. So inside parasitology laboratory, there is 2 types of the procedures takes place for the identification of the disease or for the diagnosis. First one is the macroscopic examination, and next is the microscopic examination. So what is macroscopic examination? Macroscopic examination means that this is a kind of the examination which can done by naked eyes without the use of microscope, right? So macroscopic examination is a kind of the examination which can be made by physically observing the sample. For example, this is a pen drive, right? So for example, this is a sample, right? So the diagnosis which is made by observing its physical appearance is known as the macroscopic examination. For example, the color of the front part is black and the back is red, right? The scent disc is appearing on the label. So this kind of the examinations, right? For example, the color of the stool is this, odor is this, the blood is appear in the stool or not. So those examinations which made by naked eyes, that comes under macroscopic, and those examinations which required microscope for the observation, that is the microscopic examination. I hope it is clear. So inside macroscopic examination, there will be the factors or there will be the parameters. First one is the consistency. First, this is your duty to note the consistency of the specimen. Either it is semi-solid, solid or liquid, right? Color of the stool specimen or other specimen, that either the normal color of the stool specimen is the dark brown. If the color is different, for example, gray color, red color stool specimen, green color stool specimen. So these points you have to mention inside the report also. Order. So the normal order of the stool specimen is aromatic, but if the order is different from normal, then it will also indicate a kind of the infection. So note down the macroscopic points also. Consistency, color, order. Look for the presence of blood or mucus, right? If the blood is appearing in the stool, then also note it down, right? If the presence of blood or mucus is in the stool, if you are able to see that the blood and mucus is present in the stool, that it may be suggestive that the patient is suffering from amoebic infection, right? That is the dysentery, amoebic dysentery. So here is the one questions for you, that which parasites can cause amoebic dysentery? So the answer is that the Entamoeba histolytica, which is a related or which is a part of amoebic, it will cause the amoebic dysentery, right? And one more important thing, you have to know that what is the difference between diarrhea and dysentery. Because as a technician, you have received the specimen, right? So you have to get the knowledge about that what is the basic difference between diarrhea cases and dysentery cases. I will ask from you in the end of the class, that what is the difference between diarrhea and dysentery. Next is microscopic examination. In microscopic examination, there are the procedures, that is saline wet mount, iodine wet mount, smear after concentration, and the stained smear. So in today's class, we will discuss about saline wet mount and iodine wet mount. So saline and iodine wet mount is a kind of the preparations which are done by using the microscope. That's why they are comes under the microscopic examination. For the saline wet mount, you have to do that. You have to take the specimen, right, and mix the specimen with a drop of the normal saline, and mix it with the applicator stick, cover it with the help of cover slip, and the smear is examined under microscope. And for iodine wet mount, you have to take Lugol's iodine on the glass slide, then mix a small portion of the specimen, and then again cover it with the cover slip and examine the slide with the help of microscope. So students, why we made two preparations, saline wet mount and iodine wet mount? So the reason is that the saline is colorless. There is no color of the saline, while the iodines, Lugol iodines, is a yellowish color. So first is difference is that without color, you are not able to easily identify the nuclear characters. That's why we need a coloring material, that is the Lugol's iodine. That's why we also perform Lugol's wet mount, iodine's wet mount also with the saline wet mount, right? Some of the laboratories perform, some of the laboratories also follow this practice, that they will use the only single slide, right? On a single slide, they will make two smears, one with saline wet mount and second is iodine wet mount on a single slide. For a patient, a single slide is used, right? Half of the portion is for saline wet mount and half is for iodine wet mount. So what are we getting from the wet mount? So in the saline preparation, or the saline preparations are used for the detection of the trophozoites, cysts and eggs, right? But iodine's wet mount is used for the identification of the nuclear characters of the cyst and trophozoites. So what are cysts, what are trophozoites? So I will tell you that the trophozoites are the parasites also, parasites itself, right? There will be the two forms of a parasite. Either it present in the form of trophozoite, either it will present in the form of cyst, or eggs of the trophozoites are also present inside the stool of an infected patient, right? There will be less chances to observe an intact trophozoite, but cyst and eggs are easily found inside saline and iodine wet mount, right? So in saline preparation, we can also detect the motility also, movement of the parasites. That's why we also use saline wet mount. So I hope it is clear to you, saline and iodine wet mount. So just discuss the some parasites. So first one is the Entamoeba histolytica. It is belong to protozoa group, right? And in the amoebic classification. So Entamoeba is a protozoa that causes intestinal amoebiasis, right? There is two technical terms. First one is the causative agent. Second is the disease. Just listen carefully. There is two technical terms: causative agent and disease. Causative agent is the name of the parasite, right? Means the organism which is responsible for the production of the disease, that is the causative agent. So the causative agent is Entamoeba histolytica and the disease is intestinal amoebiasis, right? So Just remember that there is two technical terms, causative agent and the disease. So causative agent is the parasite itself, and the disease is that the disease is produced by that particular parasite. So it causes intestinal amoebiasis as well as extraintestinal manifestations also. It is transmitted by ingestion of the amoebic cyst, right? And from where we can ingest these cysts, so we can ingest or we mistakenly ingest these cysts through contaminated water or food, right? So Entamoeba histolytica is transmitted by fecal oral route. All right. So this is a trophozoite form of the Entamoeba histolytica, as you can see in this picture, that this is the Entamoeba histolytica. This is the pseudopodia, which is also present in the amoeba, as this belongs to amoeba group. That's why they also form false feet. That is the pseudopodia. There is a food vacuole present inside the Entamoeba histolytica, in which it stores its food. This is the cytoplasm, and there are also some ingested RBCs, right? As you know that it is also living inside the intestine of the patient, of the person. So it ingests the RBCs. So RBCs will remain intact after the ingestion, and this is their chromatin and karyosome, in which is present inside the nucleus of the Entamoeba histolytica, right? It is a unicellular. So just see the life cycle, how it is reached in our intestine, right? What is the life cycle of Entamoeba histolytica? So just see, we ingest the food which is contaminated with the cysts of Entamoeba histolytica. First step is the ingestion. Then after ingestion, they will move down towards the intestine, because along with the food, they will reach inside the intestine. In the intestine, they will start multiplication, right? And they will form their trophozoite form and also form cyst, right? And start migrating towards the other organs. Sometimes it will also affect the liver, lungs, and if the infection is more serious, then it will also affect our brain, right? The trophozoites also migrate towards the brain if the infection is untreated. Then we will start appearing in our feces if we have the infection. In the feces, either cyst will appear. It may be either mature or immature cyst, or intact trophozoite is also present inside the stool specimen. So this is the life cycle of the Entamoeba histolytica, right? Second parasite is Giardia lamblia, which belongs to, write in the chat box, it is belong to which group? Amoeba, ciliate, flagellates, or sporozoa? So Giardia belongs to flagellates, right? So Giardia is a single cell parasite. It causes diarrheal diseases, which is known as giardiasis. So the disease caused by the Giardia is the giardiasis, and it includes small flagellates also that can live in the digestive system of the various animals like cat, dog, termites, rat, and sometimes in the humans also. Giardia is found on the surface of the soil, mud, or contaminated water, right? So it is also transmitted to humans by fecal oral route. For example, you can see in this diagram, contamination of food, contaminated food or water is ingested by a person, right? Then the trophozoite, the cyst will entered in the patient's body along with the food, and exstation will occur. Means exstation will occur means the trophozoite will start appearing in the intestine, right? The cell wall of the cyst is getting ruptured and the trophozoite is released from the cyst, and then they will multiply, right? They will multiply in the gut and produces cyst also, And the cyst will also appear in the stool specimen, which specimen is sent to the laboratory for the diagnosis if the patient feels symptoms. So it is about the Giardia. Next parasite is the Plasmodium, right? This is very most common infection which affect the India every year. That is malaria. It can cause malaria. So malaria is caused by a parasite which belongs to the Plasmodium species. So it is a mosquito-borne infection, and the species which is responsible for the malaria, that is the Plasmodium. And just see the life cycle of the malarial parasite, that is the Plasmodium parasite. The first one, the mosquito, which is okay. You can write down the causative agent of the malaria. Which mosquito is responsible for the uninfected female Anopheles will bite an infected patient, then it will suck the blood, right? And after that, the male and female gametes will enter inside the mosquito, and again formation of egg take place inside the mosquito, and the mosquito is infected with the infected patient from an infected patient, and then again this mosquito will bite another patient. That's the life cycle of a malarial parasite, which can continue. So first is transmission of the sporozoites to the human. Second, sporozoites enter the liver cells. After entering inside the liver and infecting liver cells, they will enter in our RBCs, right? And then the cycle will take place inside the erythrocytes. Then they will form their sexual cycle, and then they will again transmit it to an uninfected mosquito, right? So just remember, in life cycle of a Plasmodium species, there will be the two phases. First one is intrahepatic phase. Second phase is intraerythrocytic phase. In intrahepatic phase, the division of sporozoite will take place inside the liver cells, and other division will take place inside the RBCs. That's why the blood smear inside when we take the blood from infected patient which having the malaria, we can see if we observe the blood smear of that patient inside the microscope, the stages of their cycle will appear inside the RBCs, right? Just see, just listen that if you receive any sample inside the laboratories, when you are going for your internship, just do one thing. If you're receiving any specimen that is the malaria positive, right? Nowadays, we can check the malaria positive or negative with the help of rapid kits, right? We add a drop of a blood and then add buffer. The test will run, and within 15 minutes, we will get the results. If the two lines are appearing on the control as well as on the test, that it means malaria is positive. If one line is appearing, it means malaria is negative, right? If you receive any patient with malaria positive, right, just take the sample, place a drop of the blood over the slide and make a good smear, and then stain the slide with the normal Romanowsky group stains, for example, either with the Giemsa or Leishman stain. Then observe this slide under microscope, right? Observe the RBCs of the infected patient. You can find that there will be the parasites are present inside the RBC, which you can easily see inside the microscope, like that. This shape, this ring form shape, either a bunch of small merozoites. These types of the shape is clearly visible under microscope. Just do this task when you are going for your internship. Next is the Ascaris lumbricoides. Ascaris belongs to nematode group, right? and it is also known as roundworm. Roundworm, in simple words, we can say it roundworm, but the scientific name of roundworm is Ascaris lumbricoides. Just note down, it is very important. In many exam papers, some of them are asked for the scientific name of roundworm. So the scientific name of roundworm is Ascaris lumbricoides, that can reside in the small intestine of human. Just see, this is the form of the male and female Ascaris lumbricoids, right? They have the mouth, excretory pore, lateral line, and female genital aperture. Male have curved tail and female have no curve in the tail. And the basic difference between male and female trophozoite is that male is shorter in length, but female is longer than male, right? To see the life cycle of Ascaris lumbricoid, it is also infected by fecal oral route. After ingestion of contaminated food and water, the patient gets the infection of Ascariasis, and then egg will enter inside the mouth. Then it will go inside the small intestine, and the eggs are then the larva will comes out from the eggs, and then it will migrate towards the lungs and the liver. And if the infection is untreated, then it will also move towards the brain also, right? And then it also start appearing in the stool specimens. Second is the Ankylostoma duodenal. So Ankylostoma duodenal is a hookworm, right? So the scientific name of hookworm is Ankylostoma duodenal. They are the blood-sucking parasites because they have the hook at the end of their tails. That's why they are the hookworms. And this hook helps them to suck the blood of the patients, of the persons, right? It is also living inside the small intestine of the human beings. And the persons which are infected with the Ankylostoma duodenal, they are suffering from the disease which is known as the helminthiasis, because it is belongs to helminth group, right? And the example of hookworms are Ankylostoma duodenal and Necator americanus. In India, the infection which is more common, that is due to Ankylostoma duodenal, right? It is entered in our body through grass. When we are walking barefoot over the grass, then if the grass having the larva of Ankylostoma duodenal, then they will penetrate through our skin, right? So do not walk barefoot over the grass, because if the grass is contaminated, then the larva will enter to your body by penetrating the skin. Then they will enter in the skin, and then from the skin they will migrate toward the intestine, and then the eggs will appear in stool also, because they start multiplication inside the intestine. They will form eggs like that. Next is Taenia solium and Taenia saginata. Taenia solium and Taenia saginata is a parasite which belongs to the tapeworm species. As I already told you, the example of roundworm, hookworm, and the Taenia solium and Taenia saginata is a type of tapeworm, right? Tapeworm is the journal name, and the scientific name of tapeworm is Taenia solium and Taenia saginata, right? So Taenia saginata is also known as beef tapeworm, and Taenia solium is known as pork tapeworm, right? Beef because it is present in cow, and pork because it is present inside the pigs. So that's why Taenia saginata is known as beef tapeworm, and Taenia solium is known as pork tapeworm. So if you want to remember the names in a most easy way, so the easiest way is that saginata. The word saginata is large, right? That's why this is present in cow. That, right? So it is a beef tapeworm. And solium is smaller than saginata. Spelling of the solium is small than saginata. So it is a pork tapeworm. The size of the pig is smaller than cow. So this is just a simple way. to remember, right? So it is infected by, it can enter or inside the humans by ingestion of the uncooked or raw meat, right? If the uncooked or raw meat or half cooked meat is ingested by a person, beef is ingested by a person, and if the cow is infected with this parasite, then there will be the chances that the person will acquire the Taenia saginata, and it will start multiplication in the intestine and and produce the symptoms. Same as in the case of Taenia solium, it can also transmitted by fecal oral route, because if the uncooked meat or raw meat is ingested by meat of pig is ingested by a person, then same cycle will repeat, right? Next concept is virology. So we will done with the we will discuss the parasitology. So next move on the virology. So as the name suggests, and as I already told you, that virology is a branch of the microbiology which deals with the study of viruses. And why we read the viruses? Because they are also producing disease to the humans, producing symptoms. So to find out the cause of the disease, to find out the preventions, to find out the treatment, or to confirm the diagnosis, we have to read about the viruses, right? So let's discuss some characteristics of the viruses. Viruses are the non-cellular microorganism. They are not living, right? They are only living inside the host. After outside the body of the host, they are non-living. They are composed, or they are made up of the genetic material, either DNA or RNA, not both. Inside the viruses, either DNA is present or RNA is present, not both genetic material is present inside the viruses. They are the smallest microorganism from all, right? They can invade the living cells and also having the proteins. So there are some general characteristics of viruses. They have no nucleus. They do not have organized cell structure. They either have DNA or RNA. They are enclosed in a protective coat, which is known as the capsid. They do not respire, do not have metabolic system, do not grow, do not reproduce, right? They considered as both as living or non-living, because they are inactive outside the host, but they are active when they are present inside the host. So the simple mechanism of the viruses is that they have nothing. They have only protein coat, that is the capsid, and the genetic material, either DNA or RNA. Rest of the same things they will use from the host. They will use the machinery of the host, right? They just simply insert their genetic material in our cells, and our cells will, because our cells are naturally multiplied, so they insert their genetic material, and our cells will work for them. The cells will multiply, and the genetic material is also multiply. So they are just make fool the humans, because they are using the machinery of the humans. They have nothing. They are inactive without the host. The types of the viruses: single-stranded negative sense RNA viruses, single-stranded positive sense RNA viruses, double-stranded RNA viruses, or DNA viruses. So there are some examples of the double-stranded DNA, double-stranded RNA, single-stranded negative or positive sense RNA, right? Next, most important topic is bacteriophage. Bacteriophage is a virus that infects the bacteria and reproduce inside it, right? So bacteriophage is a virus that infects the bacterial cell and start reproducing inside it. They vary a lot in their shape and genetic material. Bacteriophage may contain DNA or RNA, either. The gene ranges from 4 to several thousands, and their capsids can be either isohedral, filamentous, or head tail in shape. They are the shapes. structure of bacteriophage. So bacteriophage consists of a head and a tail. Just see in the picture. A bacteriophage has a head and tail, right? This is the head. Inside the head, the nucleic acid or nucleic material is present, or the nucleic material is encoded inside a protein code which is known as capsid. This is the collar, this is the tail tube sheet, this is the base plate, and long fiber tails, and there are the spikes. So this is the bacteriophage. Okay. So thank you, students. It is all about the overview of parasitology and virology. And for detailed information, just visit your LMS and see the unit-wise videos which are available on the